It looks like the stand is working. I have already outlined a lot of chemistry, but I'm going to do it again to get the repetition and experience since it's a challenging subject. So, without further ado, general principles. There are 3 energy systems in the body: aerobic, oxygen; anaerobic, not oxygen; and phosphagen, like phosphates. The ETC, electron transport system, and Krebs, located in the mitochondrion, are aerobic, oxygen-based. Glycolysis, cytosol, is anaerobic, not O2. Anaerobic metabolism is faster, but produces less ATP. The phosphagen energy system uses high-energy phosphate compounds like ATP and ligase-mediated reactions. Just checking to make sure we're recording and everything's good. Okay. Chemical reactions. An enzyme is a protein catalyst which speeds up the rate of chemical reaction by lowering the activation energy without being altered. Enzymes are not consumed by the reaction they catalyze, nor do they alter the equilibrium. Enzymes do not change the delta G, or Gibbs free energy, which is the energy needed to do the work. A proenzyme is also known as a zymogen. It's an inactive enzyme precursor, which requires a biochemical change to become active as an enzyme. An apoenzyme is the protein component of an enzyme to which the coenzyme is attached. Enzymatic reactions are affected by substrate concentration, pH, and temperature. Oxidation is the process in which there is a loss of hydrogen electrons or a gain of oxygen. Oxidase is an enzyme that causes oxygen in a compound to be changed to water. Dehydrogenase is an enzyme which oxidizes a compound by removing the hydrogen. Reduction is a process in which there is a gain of hydrogen electrons or a loss of oxygen. Reductase is an enzyme which adds hydrogen. Carboxylation is a chemical process by which a carboxyl group is added or displaces a hydrogen atom. Carboxylase is the enzyme which catalyzes the addition of a carboxyl. Decarboxylation is the process where the carboxyl group COOH is removed from an organic compound like CO2. It's commonly replaced by hydrogen. Decarboxylase is the enzyme that catalyzes the release of CO2 from compounds. Hydrolysis refers to the cleavage of a compound by the addition of water. The hydroxyl group being incorporated in one fragment and the hydrogen in the other. Hydroxyl one, hydrogen in the other. Hydrolase is the enzyme that facilitates hydrolysis, as in sucrase in the breakdown of sucrose. Phosphorylation is the process of introducing a phosphate group into an organic molecule. Phosphorylase is the enzyme which adds inorganic phosphate to a substrate without ATP. Phosphatase is the enzyme that removes a phosphate group, removes a phosphate from its substrate like G6-phosphate and converting G6-phosphate to glucose. Kinase is an enzyme that moves a high-energy phosphate group from a donor, like ATP. Isomerase is an enzyme that changes the configuration, but not the composition, like G6-phosphate to fructose 6-phosphate. Mutase is a type of isomerase which moves one chemical group to another part in the same compound movement. For example, the mutase that moves phosphate from C3 to C2 in 3-phosphoglycerate to 2-phosphoglycerate. Transferase is an enzyme that catalyzes the transfer of a functional group from one molecule to another, as in aminotransferase, which transfers NH3 from one molecule to another. Ligase is an enzyme that catalyzes the joining of two molecules. DNA ligase joins DNA fragments. Km, aka Michaelis-Menten constant, is the substrate concentration required to reach half of Vmax. Vmax refers to the max velocity of a reaction. It's proportional to the enzyme concentration. An inhibitor is a substance that slows down the rate of reaction of an enzyme. 3 ways of enzymatic regulation: non-competitive inhibition, competitive inhibition, and allosteric regulation. In competitive inhibition, the inhibitor binds to the same active site as a normal substrate without undergoing a reaction. It's reversible. Competitive is reversible, same active site. Increasing the substrate concentration will overcome the activity of the inhibitor, thus Vmax will not change. Non-competitive inhibition. The inhibitor always binds to an other site instead of the typical active site. It's irreversible. It reduces Vmax. However, increasing the concentration of the substrate will not affect the activity of the inhibitor. In allosteric regulation, an enzyme's activity is regulated by binding an effector molecule at the enzyme's allosteric site, a site other than its active site. Effectors that enhance the protein's activity are referred to allosteric activators. Those that decrease the protein's activity are allosteric inhibitors. All right. Energy compounds. NADP, I still have never said this fully through. Nicotinamide adenine dinucleotide phosphate. NADP is the phosphorylated form of NAD. NADP is used in anabolic reactions like lipid and nucleic acid synthesis and the pentose phosphate pathway, which require NADPH as a reducing agent. NADH is the reduced form. It's given off, NADH is given off in Krebs to make ATP. Both NAD and FAD facilitate the transport of electrons from Krebs to ETC. There are 4 basic energy fuels of the body: carbs, proteins, fats, alcohol. Carbs are 4 kcal, proteins are 4, fats are 9, alcohol is 7. The alcohol calories are empty, they have no nutrients. Catabolism of energy fuels heat generation and ATP synthesis. ATP, adenosine triphosphate, is the universal energy currency of the body. Catabolism is a degradative process. Breaks down large molecules into smaller ones, releasing useful energy. Anabolism is a biosynthetic process, building up which constructs large molecules from smaller units. These reactions require energy. Types of chemical bonds. Peptide bond is covalent. It's formed between two molecules when the carboxyl group of one reacts with the amino group of the other, thereby releasing a molecule of water. This dehydration synthesis is also known as condensation, usually occurs between amino acids. Hydrogen bond results from dipole-dipole force when a hydrogen atom is bonded to nitrogen, oxygen, or fluorine. Nitro, O2, or fluorine. Highly electronegative atom. H-bonds are chemical and found between the bases in DNA and RNA. An ester bond is chemical covalent between glycerol and a fatty acid. Esters consist of an inorganic or organic acid in which at least one hydroxyl is replaced by an O-alkoxy group. Glycosidic bond is the chemical bond between two sugar molecules. Phosphodiester is the chemical bond between two nucleotides. It's a group of strong covalent bonds between the phosphorus atom and a phosphate group and two other molecules over two ester bonds. I'm going to have to study the phosphodiester more. I cannot picture it at this moment. Generalizations about metabolic pathways. What is the purpose of the pathway regarding either the substrate or the product? What's the tissue location of the pathway, where it's most important? What's the cell site? Where in the cell the pathway occurs? Cytosol, mitochondria, both. Mitochondria fatty acid oxidation, Krebs, and oxidative phosphorylation. Cytoplasm glycolysis, fatty acid synthesis, HMP shunt, and protein synthesis. Both heme synthesis, urea cycle, gluconeogenesis. I read that really weird. It's going to be hard to follow. Mitochondria is fatty acid oxidation, Krebs, and oxidative phosphorylation. Cytoplasm is glycolysis, fatty acid synthesis, HMP shunt, and protein. Both mitochondria and cytoplasm, heme synthesis, urea cycle, and gluconeogenesis. I should probably have looked up more before making this video. Okay, next section. Sequence of events. What's the overall sequence and number of stages or reactions? What are the key steps that form control or the rate limiting points for main branching points? Rate limiting step is a crucial step that controls how fast or slow the pathway goes. For example, in glycolysis, it's phosphofructokinase. In gluconeogenesis, it's fructose 1,6-bisphosphate. In Krebs, it's isocitrate dehydrogenase. Glycogen synthesis, glycogen synthase. Glycogenolysis, I hope that's the right pronunciation, glycogen phosphorylase. HMP shunt, glucose 6-phosphate dehydrogenase. Urea cycle, carbamyl phosphate synthetase. Fatty acid synthesis, acetyl-CoA carboxylase. Fatty acid oxidation, carnitine acyltransferase. Cholesterol synthesis, HMG-CoA reductase. That was a very long video. I'm going to go to the next one.